Dear children, in the last modules, we had learned about the four different types of biomicromolecules, which are the lipids, the nitrogen bases, the amino acids, and sugars. And in this module, we are going to begin with biomacromolecules. So, who are these biomacromolecules? They are biomolecules which are having the molecular weight greater than 1000 daltons. And the different types of macromolecules are the proteins, the polysaccharides, and the nucleic acids. And as I told, their molecular weight is greater than 1000, that is around 10,000 daltons and above. And they belong to the acid insoluble fraction, that is the retentate. And it includes the macromolecules which forms the cytoplasm and organelles, that is the biomicromolecules. They constitute the cytoplasm, whereas the macromolecules they include the macromolecules from cytoplasm and the organelles. Whereas there is a difference with regard to the lipids. Lipids is not strictly macromolecules. Even though we had learnt it in the micromolecules, they are not macromolecules. They gain a intermediate structure. Let's see why. Because their weight does not exceed 800 daltons. Thus, they belong to biomicromolecules. But it comes under acid insoluble fraction. Whereas micromolecules, they are acid soluble fraction, and the macromolecules, they are acid insoluble fraction. But the macromolecules, they should have a molecular weight greater than 1000 daltons. That is also not getting satisfied over here. It's having a molecular weight of only 800 daltons. But they belong to the acid insoluble fraction because many lipids they are arranged into structures like cell membrane. So we know cell membrane is acid insoluble. And when a tissue is grinded, the cell membrane are broken and it forms water insoluble vesicles. So they cannot be filtered along the acid soluble fraction. But they do not belong to the acid insoluble fraction also. So they gain an intermediate structure. So the average composition inside the cell of water is about 70 to 90%. Protein, it constitutes about 10 to 15%. Carbohydrate, around 3%. Nucleic acids, 5 to 7%. And ions, it's about 1%. So let's begin with the first biomacromolecule, that is proteins. Proteins are polymers formed of amino acids. They are not homopolymers, they are heteropolymers formed of different types of amino acids. So they are called as heteropolymers of amino acids. And they are called as polypeptides. Why they are called as polypeptides? Because amino acids, they are linked together by a peptide bond. Okay, so the COOH, the carboxyl group of one amino acid, it is getting linked to the amino group of the adjacent amino acid, forming a peptide bond. So many peptide bonds join together to form a polypeptide chain. So peptide bond is formed when the carboxyl group of one amino acid reacts with the amino group of the next amino acid by releasing a molecule of water. Okay, by releasing a molecule of water, a peptide bond is formed. And many peptide bonds together forms a polypeptide chain. So the proteins have diverse functions inside a cell. It helps in growth and tissue repair. It helps in the transportation of substances. For example, the GLUT4 protein, it enables the glucose transport into the cell. It allows the entry of glucose into the cell. And it is acting as a ground substance. It is supporting the entire, it is acting as a supporting substance inside cell. For example, in the case of collagen fibers. It acts as antibodies to fight against the infectious agents entering into our body. It acts as a receptors, for example, receptors for smell, taste, and hormones. Some of the hormones, they are protein in nature. For example, insulin. Some proteins are enzymes. Okay, example is trypsin. Some proteins, there are some pigments, they are proteins. For example, hemoglobin, etc. So proteins are major structure of hemoglobin. Then it also forms a part of enzymes and also hormones. So most abundant protein in the animal world is collagen, and that in the plant, on the total biosphere. In the total biosphere, the abundant protein is a rubisco. What does rubisco stand for? It stands for ribosome. ribulose, bisphosphate, carboxylase, oxygenase. Okay, 1,5-bisphosphate, carboxylase, and oxygenase. And we had learned in the biosynthetic pathway of photosynthesis. And structural level of proteins. It has different structures: primary structure, secondary, tertiary, and quaternary. So these are the levels of organization of proteins. So let's begin with the primary structure of proteins. It describes the sequence of amino acids, a number of amino acids joined together by a peptide bond. So this is nothing but a sequence of amino acids which constitutes a primary structure. So it will have two ends. One is called as the left end, the other is called as the right end. The left end is called as the end terminal. Why? In this here we can see the amino group in each tree. Okay, so that's why it's called as the end terminal. And as the other end is called as the C terminal. Why it's called as the C terminal? Because we are having a carboxyl group. So a number of amino acids which are joined together by peptide bonds, with one end having the C terminal and the other end having a end terminal, is called as the primary structure of proteins. Whereas secondary structure, one or more polypeptides chains, they are folded. They folded to form a helical structure. So when one or more polypeptide chains, they are folded in such a form, it forms a helical structure. It's called as a secondary structure. So it is having only right-handed helix. Right-handed means it is folded towards the right-hand side. Okay. For examples of the secondary structure of the right-handed folded helix is keratin fibroin, that is a silk fiber obtained from the Bombyx mori. Okay. So silk fiber, fibroin, all these are examples. Keratin, which is found in hair. All these are examples of the secondary structure. So you can see this is a structure of hair and this is a structure of the fibroin. Okay. So they are two or more polypeptides which are bounded to form a helical structure. Third is the tertiary structure. Okay, here the helical polypeptide chain is much more folded. Okay, they are much more folded to form a hollow woolen ball. Okay, and it gives a 3D structure to the proteins. And tertiary structure is necessary for many biological activities of proteins. For example, myoglobin, enzymes. So all these proteins, they have tertiary structure. And last is the quaternary structure of proteins. Here, more than one polypeptide chain that forms tertiary structure, and each chain functions as subunit of a protein. Okay, more than one polypeptide chain, it forms a tertiary structure, and each chain it acts as a particular subunit of protein. For example, hemoglobin. Hemoglobin is having 4 units. That is 2 alpha and 2 beta subunits are present. See, this is a 2 alpha and 2 beta. Okay, and it's having a tertiary structure, and each subunit over here is acting. Okay, each chain functions as a subunit of protein. So that's about the quaternary structure of protein. So proteins are the polymers, heteropolymers of amino acids which are joined together by peptide bonds. And there are 4 levels of organization: the primary, secondary, tertiary, and quaternary structure of proteins. Thank you.